As scientists, we like asking questions. But be honest with me. Have you ever regretted asking a question? I have, multiple times. For example, the other day, I was planning to have a pizza with my sister. And just to be on the safe side, I asked her which topping she prefers, mushrooms or olives. Now, I was already at the restaurant. I didn't have much time before order, and I was expecting a short, specific answer, one word actually. Instead, I got about 20 video messages. Don't get me wrong, I love my sister, and I'm sure that there is plenty of important information in these videos. But right now, I was interested just in a pizza topping. And this information is somewhere there, but it's very hard to retrieve. I asked my sister a simple question, and I expected to receive a simple answer. This is actually what happens in big data analysis. So now imagine that you're talking to big data. You're also asking a specific question. What is the difference between A and B? You get a response, but this response is heavy, highly unspecific, and hard to interpret. So what can you do? This is exactly the challenge I'm facing in my PhD. The data I'm talking to is in the field of Alzheimer's disease. I'm a molecular biologist, and I'm studying the biology behind the aging brain. This topic is particularly relevant today because of its clinical implications. While all of us age, only for some of us, aging brings cognitive decline. 55 million people today have dementia, and this number is expected to grow in the following years with increasing life expectancy. Alzheimer's disease contributes to the majority of dementia cases. So it's $1 million question. What is the difference between healthy brain cells and cells from Alzheimer's disease brains? This is the question I'm asking in my PhD. Fortunately, due to the development of biotechnology, I also have the data that can answer this question. If I may even be so bold, today, biology is data science. A typical data set I'm working with contains a few dozens of brain samples. Each brain sample includes thousands of cells, and each cell gives a reading of tens of thousands of molecules. Now multiply these numbers, and you will get an approximate size of the data set a molecular biologist has to deal with. So we arrived at our original problem. We have tons of information, a huge data set, and we want to retrieve a specific response about the difference between healthy brain cells and Alzheimer's disease brain cells. Here, my conversation with the data begins. To understand it, let's take an example from a different world. I'm going to read a poem, and I want you to think and decide when it was written, in 17th century or in 20th century. When most I wink, then do mine eyes best see, for all the day they view things unrespected. But when I sleep, in dreams they look on thee, and darkly bright are bright and dark directed. Raise your hands those who think it was written in 17th century. Okay, and in 20th century? Okay, but the majority was right. It's a 43rd sonnet by William Shakespeare. Now, those who are right. I claim that you didn't really need the whole poem to decide, but you mostly relied on a few specific words. Can you name them? The and mine eyes, right? So, indeed, these words suggest that this poem is quite old, whereas all the other words could likely be in modern poems as well. Now, if we translate your intuition to data, we can just count how many times every word appeared in the poems of 17th century and of 20th century, and see that mine eyes and thee have the biggest gap between the two. So we can name them 17th century words, whereas all the other words, we can say that they're words used in both centuries, right? This is the basis of the approach I'm taking in talking to big data. Of course, I'm not doing it manually. I'm using an algorithm. This algorithm is called information bottleneck because it squeezes the original information through a metaphorical bottleneck, leaving only the relevant information behind. So its first output would be a simple division of all the words in all the poems to those that are used only in the 17th century, only in 20th century, and used at all times. Now, let's go back from poetry to biology. It's not going to be so hard because, in fact, poems and biological cells are not that different. Just like a poem is built of words that define its meaning, a biological cell can be viewed just as a set of molecules that completely identify its state and behavior. Ironically, molecules I'm working with are even denoted with the combinations of letters, just like words. This family is called RNA. Just like when reading a poem, every word gives more of your understanding of the whole story of the poem, every RNA you find in a cell tells you more about what the cell is going through and how it behaves. But just like not all the words are informative about the century the poem was written in, not all the RNA molecules change between healthy brain cells and Alzheimer's disease brains. So we can use the same algorithm. I'm dividing all the RNA molecules in the dataset to those that are expressed specifically in Alzheimer's brain cells, those that are expressed in healthy brain cells, and those are not affected by the disease. So it might seem like we're done now, right? We have our molecules that are specifically expressed in Alzheimer's, so we have the answer to our question. I have to disagree. Sometimes putting all the important elements in one place is simply not enough. For example, imagine that I'm saving all the important files on my desktop, because there they will be easiest to find, right? In the end, my desktop looks like this. All these files are important, but it's impossible to retrieve a specific file. It would be much easier to organize these files into thematic folders. This is exactly what information bottleneck does with a list of RNA molecules. It divides them to specific folders that now represent biological processes that involve the molecules inside them. Now this list of folders is much easier to interpret. Now let me give you a glimpse at my actual results. Okay, just kidding. But these are the results, and if you plot them, it actually looks much easier to interpret. It gives you a hierarchy of genes, and the first division, as I said, are RNAs in Alzheimer's brain cells, unaffected RNA, and RNAs in healthy brain cells. It further divides them to thematic folders that now represent biological processes. Let me open some of these folders and explain the meaning behind them. The first folder, which is associated with the highest risk of Alzheimer's, represents cellular death. It means that cells in Alzheimer's brains are dealing with such immense stress that it eventually kills them. Another folder represents molecules that are involved in the process that reduces the activity of the brain cells. Reduced cell activity in the brain can explain many of the symptoms of Alzheimer's, like cognitive decline. And note that it is further connected to the cell death folder in the hierarchy, which means that there might be some connection between the two processes. On the other hand, healthy RNAs are divided into a process of memory. So metaphorically speaking, we can say that a healthy cell has more storage available for making new memories, whereas an Alzheimer's cell is limited in that ability. Another last process is stress response. We can speculate that healthy cells have a strategy to deal with stress, whereas Alzheimer's cells don't, and that's why it harms them the most. Now, let's summarize what we did in the last 10 minutes. I showed you how you can talk to big biological data using information bottleneck algorithm. We first divided the molecules to relevant and irrelevant ones, and further divided them to more specific categories representing biological processes. We even opened some of the folders and saw what they actually represent. This reflects a computational part of my PhD, where I'm adapting information bottleneck algorithm for use in single-cell RNA sequencing data. In parallel, I'm also conducting experiments in the lab to validate my data analysis and to construct new data sets to study other aspects of aging brain biology. So I hope I convinced you that compression and filtering are very important, and sometimes less information is more informative. But there are a few exceptions. For example, if I filter my sister's messages, whoa, she will never forgive me. With that, I thank you. I thank my sister for cooperation, and I would love to take questions.
שומעים? שומעים?
Can you hear me?
פחות מבלי מיקרופון נראה לי.
Hello? Okay.
Okay.
So, do you only categorize and filter the RNAs that you find in the different cells, or do you already have it like with categorized also by the biological functions that may be different, or the possible treatments maybe that are known for
The
certain
RNAs?
problems?
The molecules or the cells?
The cells.
So I assume in order to use the algorithm, I need to have the labels of the cells. So yes, I have to know which cells are healthy and which cells are Alzheimer's. And my unknown that I'm discovering are the genes, are the molecules. So is that the right direction of the question? Yes, I could. Yeah, totally. I could, for example, I could create the groups by combinatoric, like, for example, this group is Alzheimer's female, this group is Alzheimer's male, and this group is, you know what I mean? So that I can combine different labels in the setting such that the molecules will be informative about both factors. Is it answering? Of the cells? So the cell type is a major signal in single cell RNA sequencing data, so I'm using the algorithm for a specific cell type if I'm studying the disease, because otherwise the only signal I would see is cell type. So, for example, the results I've shown you is for excitatory neurons specifically. But I could also run it on microglia, astrocytes, and other cell types, and then I would see different hierarchies of genes and different biological processes that are involved in this specific cell type and the disease progression. Thank you.
Hi, Oriane from Tel Aviv University. That was really amazing. Thank you.
Thank you.
And maybe what you said now about cell types answers my question, but I wanted to ask, what if some RNA sequences are participating in the different folders?
That's a great
what
question.
do you do? And maybe what happens if suddenly you discover that something you thought is in one folder actually belongs in several?
So it's a great question, and it does happen, but the output is probabilistic. So for every molecule, I have a list of probabilities of assigning this molecule to every folder. So it's actually a very interesting result if I have an RNA, because it does happen in biology that one gene can be involved in many processes. And yeah, this is one of the outputs it can give. So.
Hi, thank you very much. It was amazing.
Thank you.
Very clear. I wanted to ask about the folders. It seemed rather manual. Is it that manual, or do you use some kind of algorithm to choose the folders for you?
To choose the folders are the output of the information bottleneck algorithm. So the hierarchy I showed before is like the plotted output. So it's basically the representation of matrices of numbers that the algorithm provides. Yeah, but recognizing the specific biological processes that are enriched in a specific folder, I use gene ontology resource, another tool in biology. It's not the output of the algorithm. The output of the algorithm is just the cluster of genes, and then I realize that these genes are all connected by a process that is known to be involving those genes. Thank you.
So I have 2 questions, and sorry again that I'm an anthropologist of Alzheimer's, but not a biologist, so I hope the questions is okay. So first of all, about your data sets, can you say a little bit more about it, and is it already people who are affected by Alzheimer's that are part of the data set? Okay, thanks. And then the other question is about the unaffected RNA. And is there a research, or is it something that you thought about, that in a sense the unaffected RNA is the mutual between people who are unaffected and
Right.
people who didn't experience Alzheimer's and people who have? So, and is there anything about that? Thanks so much.
Thank you. Thank you for these questions. I think it's amazing that I managed to to get questions from different fields. Thank you. So about the data set, yes, it's a data set of 48 individuals, post-mortem brain samples. 24 of them are diagnosed with Alzheimer's when they were alive, and also have the pathology of the Alzheimer's that was discovered after their death. And 24 are controls, age-matched, so they're also elderly, but they're healthy cognitively. And for your second question, so unaffected RNA, there's a lot of RNAs that are involved in basic cellular functions, and they're not affected by the disease. And it's always like that when you analyze biological data. A specific condition, specific stress only affects a part of the processes the cell is involved in. That's a great question. I didn't look at those, so it's a great question, and it's definitely interesting to look at that. And it also, like for example, the orange and the pink folders are the mutual ones, and you can look at them and also see what processes they represent. And sometimes it's surprising because maybe it has a process that you thought would be affected and it's not. Yeah, but it's definitely interesting too.
Thank you very much, Sima.
Thank you.
Thank you.